Thank you all for giving me the opportunity to talk about some of my lab's research. So my name is Todd Freeberg. I'm a professor in the departments of psychology and ecology and evolutionary biology at the University of Tennessee. And today I want to talk about, it's maybe an odd title, Songbird Social Psychology, Flock Complexity and Chickadee Behavior. So I'm going to mainly focus on one older study and a couple more recent studies that starting to change the way I think about social groups and the species that we study. And let's get right to it. So the outline is to give you a little background to the projects that I'll talk about, and then go over these three studies. This older one looking at the effects of group size on calling behavior. Second one looking at the effects of group familiarity on calling behavior. And then the diversity of mixed species groups and how that relates to a non-communicative behavior, foraging success, and a novel feeder task. So just a quick background to the work. One of my heroes in this field of animal behavior is Niko Tinbergen. And in answer to the question, like, where did you get this title, Songbird Social Psychology? Well, hear me out here. So Tinbergen, as hopefully we all know, argued decades ago that in behavioral studies, there's basically four different kinds of questions that we can ask. So these levels of analysis, or what are sometimes called Tinbergen's 4 why questions. So two of these are ultimate questions having to do with populations. And so these are questions having to do with phylogeny and questions having to do with selection and fitness related outcomes. So adaptiveness or adaptation questions. Then the other two levels of analyses are more focused on the individual, the proximate questions we can ask about developmental factors, so the roles of experience, maturation, etc., on the production of a behavior, and then immediate causal mechanisms, so mechanistic questions. And social psychology is interesting in this context because it sort of is inherently integrative for these levels of analysis. It's focused on mechanisms, so how the current social context influences your behavior, but it also bridges really nicely, I think, to adaptiveness kinds of questions of how variation in social contexts can influence individual behavior over generational time. A great example of this, I'm actually sorry now that I haven't included this, is Median Andrade's recent advances in the study of behavior on the role of social context in fitness outcomes in different spider species that she studies. I think that was a paper that came out in 2019. And so you might be thinking, still, I don't get why social psychology. So social psychology today, maybe particularly in non-North American countries, is the study of human behavior. But if you look back at the history of this field, so for example, this is the inner title pages of the Handbook of Social Psychology that Carl Murchison edited in 1935. So you see in the table of contents the kind of typical things you might see today in a social psychology handbook. So attitudes, sexual behavior and social psychology, age, etc., the influences of those factors on behavior. But if you look at other parts of the table of contents, you see something really interesting. It's interesting to me anyway. So 85 years ago, we were thinking about population behavior of bacteria as a social psychological question. Plant behavior, this interestingly enough has come back into the forefront of thinking about whether organisms without central nervous systems can nonetheless exhibit behavior like relationships with their environment. Insect societies, bird societies, mammalian herds and packs, right? There's some major papers in here by really seminal figures in the field. W.C. Ali, Scheelderup Ebb developed the idea of dominance hierarchies and how to measure them, right? So decades ago in social psychology, it wasn't unheard of to think of social psychological influences on non-human animal behavior. And I think it's worth revisiting that, actually. So social psychology is the study of how individuals are affected by their immediate social environment. And there's just a plethora of examples of this in the recent and not so recent literature. So Katie Slocum's study of chimpanzees that modify their recruitment screams after an agonistic conflict kind of interaction. But that depends on the audience. So if there's a more dominant individual in the nearby environment and they've just been aggressed, they call differently than if a dominant individual is not around. It's not just in non-human primates, right? So male head bob display structure in a neotropical lizard, relation to social context, right? So animals are head bobbing differently depending on who's in their immediate social environment. And like I mentioned, there's a huge range of these factors. So they go by different names: audience effects, social facilitation, local and stimulus enhancement, herding, synchronization, coordination, cooperation, and so forth. These are all social psychological influences on behavior. So I've been studying for a couple decades now, Carolina chickadees. Pusili carolinensis. This is a very small songbird species that lives in the southeastern United States. University of Tennessee, where I am, is here in the eastern part of the state of Tennessee. This is my hand holding one of these birds. They're quite small. And they're members of the Paridae. So this comprises chickadees, tits, and titmice throughout the world. And one reason I got interested in studying these species is that most of them are pretty complex in terms of their social organization. So from the spring through the summer, they do what everybody else does pretty much. They form breeding pairs, raise young, etc. But then in the late summer, most of these species form stable flocks, oftentimes of unrelated individuals. Oftentimes those flocks defend their territories from other flocks. You get, in some species, really rigid linear dominance hierarchies. In other species, it seems to be more egalitarian. Interestingly, some species seem to form relatively stable mixed species flocks. So these are quite complicated social structures in these species. The other reason I got really interested in Carolina chickadees and related species has to do with one of their major calls. It's used by both males and females throughout the year for basically functions in social coordination, social cohesion. It's called the Chickadee call. At least in North America, it's called the Chickadee call. And one reason I was interested in this is that it's one of the more structurally complex non-language communication systems that's known. So this is 9 different pictorial representations of these calls. So these are spectrograms. Frequency is on the y-axis, time is on the x-axis. And in this American Scientist article we wrote a number of years ago, the editors color-coded the different note types in these calls. And so basically, a particular note can be present or absent in any call. If it's present, oftentimes it can occur more than once. For example, here's several of these D notes, several C notes in these 2 figures. And the result of all of these note ordering considerations, including rules of note ordering, is that you have basically an open-ended system just with note composition, let alone there's acoustic variation within note types that adds even more complexity to the call system. And this complexity is not random. Structural complexity of calls is not random. So this is just a series of studies that we reviewed up to 2012 when this came out, that looks at variation in the Chickadee call and how that variation maps on to various sort of state features of individuals, as well as environmental considerations and behavioral considerations. Right, so basically that variation in the structure of the calls does not seem to be random, but seems to potentially communicate a really wide range of information. At the time of this review, almost all of the studies described here had been done with North American parrots. Luckily, that is rapidly changing, and there's an enormous amount of work coming out of European labs as well as Asian labs And one of the people at the forefront of this is Toshi Suzuki, doing a lot of work on Japanese tits and great tits. Really interesting work. Okay, so that's a quick background. So there's some relationship perhaps between social complexity in chickadees and their vocal complexity. That's one of the things that I wanted to test. And this became known, we helped develop this, Terry Ord and Robin Dunbar and I helped develop this idea called the social complexity hypothesis for communicative complexity. And the basic argument here is that a lot of social species, they vary a lot in terms of the complexity of their social groups. And that's measured any number of ways: the size of the group, the density of individuals in the group, the diversity of roles within the group, social network connections and linkages within groups, and so forth. Communicative complexity is typically measured in one of two different ways. One is the overall repertoire size of the communicative system. And the other one comes out of information theory approaches to communication. And that's like the binary digits, the bits of information in a signaling system. And what I wanted to test, and what I realized I could test experimentally, is whether variation in social complexity actually does drive variation in communicative complexity. And this had been in the literature for quite a while in a comparative sense, in a phylogenetic sense, where in ground-dwelling sciurid species, in bats, in non-human primates, key measures of social complexity, so number of social roles, colony size, group size, were strongly associated with key measures of vocal complexity: number of alarm calls, information theory, isolation call complexity, vocal repertoire size. These are, this is one of the key data sets from Karen McComb and Stuart Semple's study of non-human primates, where controlling for phylogeny, increases in group size across these 40 plus different species are associated with increases in vocal repertoire size. Like I mentioned, I've realized as I took the job at University of Tennessee that I could actually test this experimentally. Nobody had done that yet, as far as I could tell. And so I did published a paper that had two components to it. One was a naturalistic observation study. So going into the field in unmanipulated flocks, just seeing if birds in relatively larger flocks, and I should point out that flocks in Carolina chickadees are fairly small. They typically range from 4 to 6 birds in our population, and they don't always travel together. So you're typically we'll see 2 or 3 birds, 3 or 4 birds as a decent sized flock moving through space. And so a naturalistic observation study, and when the answer to this was affirmative, we did an aviary study where we actually manipulated flock size. So first, the field study at 30 different sites in eastern Tennessee. I recorded calls of birds that I naturally found either in relatively small groups, a single bird or just a pair, and compared that to relatively larger groups of 3 or more individuals. At the time, we were using an 8-note type categorization scheme that came out of Laurie Bloomfield and Chris Sturdy's lab at the University of Alberta. Got over 100 calls per site, so these were pretty large samples. The calls grade into one another, the note types of the calls grade into one another, so we don't have perfect reliability in note type coding, but we have pretty high reliability. So even though there's enormous variation and there's gradation in the different note types, it is pretty straightforward to code these different note types. So what did we find? Well, we used an information theory approach here looking at uncertainty. So greater uncertainty is associated with more diversity in the way these notes are put together to make calls. So greater uncertainty, greater complexity. And so we looked at it in 2 different measures of uncertainty that are commonly looked at. One is just the diversity and use of the different note types. And then the other one is the diversity and use of different ordered pairs of note types. And what we found for both of these, actually, I think this first one here was just a statistical trend, but for the second one was a statistically significant difference where birds in larger flocks are using calls with more uncertainty, so greater diversity of note types and ordered pairs of note types, compared to birds that are either by themselves or in just a pair moving through their territory. So that set up the experimental part of this. So I did an aviary study where I manipulated social complexity. I manipulated group size. And so I set up groups that mapped onto natural variation that you see in flock sizes for Carolina chickadees, 2 or 4 or 6 birds, captured birds from the same field site, put them in the aviaries for 3 to 4 weeks, tried to balance the number of females and males as best we could to map onto natural sex variation in the flocks. Let the birds acclimate to the aviaries and to our presence in the aviaries, and then just recorded their calls. And at the end of the study, we released them back at their site of capture. And what did we find? What we found is that there were significant effects of group size on uncertainty in these calls for both of these uncertainty measures. So diversity and use of different notes, there's a significant overall effect. And when you look at the contrast, it's birds in these intermediate group sizes that have a greater diversity of note types than birds in the small group size. If you look at the diversity of ordered pairs of notes, there's a significant overall effect, and it's birds in the largest group size that are using the most diverse, most complex differences in ordered pairs of notes in comparison to those two smaller groups. And so using group size as our measure of social complexity, and then this uncertainty information theory approach as our measure of communicative complexity, we found that individuals in larger groups or individuals put into larger groups were using more complex calls. And I think this, to this day still, I think this is the first experimental evidence to support that social complexity hypothesis for communication. Okay, so then let's shift to the question of relationships among individuals in groups and the question of familiarity. The study I just talked about had some serious limitations to it. For one, statistically, it was a pseudo-replicated study. So I treated individuals from the different social groups as being independent of one another, and that's not the safest thing to do statistically. Furthermore, group size is not the only metric of social complexity, even though that's one that's used very commonly in the literature. And also, I couldn't separate out group size from density in the study. So these aviaries that we use are all roughly the same volume. And so a larger group is not just larger, but it's also more dense in space. Tori Bergman and his colleague Biener published a study several years ago that argued that a better metric of social complexity is to think of the number of differentiated relationships among individuals. So maybe it's not so much the size of the group, but it's the nature of the relationships among individuals in the group that's a better way of thinking about complexity. And so thinking about like networks and the egalitarian structure or the linear kind of despotic dominance hierarchy in a group, maybe that tells us a lot more about the complexity of the group than simply group size. Furthermore, the study that I just finished talking about just looked at ambient calls. These were calls birds were just producing in whatever sort of motivational states, behavioral states they were in at the time of recording. I didn't really know what the functions were of those calls and that variation in those calls. I didn't really assess that in any sort of experimental way. And so I skipped over this. This is a figure from that paper on social complexity hypothesis for communication, looking at, there's different ways of thinking about complexity of groups. There's not just the size or the density of the group, but the number of social roles, the egalitarian or network structure and so forth. So we had the opportunity to work with a student from the University of Rennes who came over to do her master's work in our lab. Brittany Coppinger had recently joined my lab at the time of this study. And what we wanted to do was see if we have groups that are exactly the same in terms of size and complexity, but we manipulate the relationships among those members by having some members be familiar. In other words, they were all captured from the same flock in the field. And then some flocks where individuals were unfamiliar. Each individual was captured from a different flock in the field. Would we see an effect on calling behavior? And so after acclimation to the aviaries, bringing the birds in, letting them get used to the aviaries, letting them get used to us as observers in the aviaries, we presented them with a series of stimuli. Some of these were control stimuli, some of these were novel stimuli, and some of these were threatening, risky stimuli related to predation. And so this is a whole set of the stimuli that we presented. One was moving. Some are predator stimuli, hawk models, cat models, another hawk, snake models, control stimuli, branches, pine cones, etc. Novel stimuli, this weird robotic thing, a cheese head. This is related to the Green Bay Packers. This is my favorite American football team. And we would present these stimuli on top of a feeder that the birds were motivated to come visit in the aviaries, and then just see what their response was. We have very high inter-observer reliabilities, both in coding the birds' latencies to call in the aviaries in response to the presentation of these stimuli, and then also in terms of the coding of the note types in these calls. And then one last thing that we did in this study is that we always release the birds in our experimental flocks. We always release them back at their site of capture at the end of the study. And for this study, I also noted when I released the birds, whether they produced a call in flight, which we can just refer to for now as a flight call. This was based on some earlier work I had done. So what did we find? So on the y-axis of this first graph is the call latency, square root transform. So call latency in seconds. And first, let's look at how the birds in these familiar flocks and these unfamiliar flocks call in regards to control and novel stimuli. Well, what we see is that birds in both groups call similarly. They call, roughly 3 seconds square root transformed after a stimulus is presented. What about the predator stimuli? Birds in the familiar flocks call quickly, right? The same sort of latency as for those control and novel stimuli. Interestingly enough, birds in the unfamiliar flocks call later, substantially later than in these other conditions. So something about being in an unfamiliar flock maybe makes you a little more reluctant to produce a call that might draw attention to yourself in an otherwise risky situation. We also analyzed the calls these birds produced under ambient conditions, so away from this stimulus presentation consideration. And what we found is that birds in familiar flocks are putting together calls that are just structurally a little different compared to birds in unfamiliar flocks. So in terms of introductory notes and C notes, birds in familiar flocks use less introductory notes and more C notes compared to birds in the unfamiliar flocks. And there's no statistically significant differences for either D note usage or total notes in a call. On release from the aviaries back at the site of capture, what we found is a statistically significant effect of flock type. Birds in familiar flocks much more likely to produce one of these flight calls in comparison to birds in unfamiliar flocks, who are more likely to either not call or call upon landing. And we think, we don't know this for certain, but we think that these flight calls may be important for social cohesion as birds are moving through the territories. And so familiar birds might be a little more motivated to produce these calls on release. So what we found is that birds showed really sensitivity to their social context. And in this case, that social context has nothing to do with group size or variation in group size or density, because there was no variation in group size or density. Familiarity influenced calling behavior. And so that points out that the nature of the relationships among individuals and groups is also really key to understanding variation in calling behavior. Some of our findings point to potential audience effects on calling behavior, and this is a topic of a lot of interest now because it suggests volitional control over calling and the possibility of strategic signaling. But we're still left with, does the complexity of social groups relate only to conspecific groups? We're treating chickadees like they exist in a vacuum and only interact with one another. Well, what about heterospecifics? What about mixed species groups? And the vast majority of my work has focused on calling behavior, but is that all that's affected by these factors that we've been manipulating, group size or density or familiarity? So that leads into the last study that I wanted to go over today, and that's looking at group diversity and foraging success. And so the question here really is, does mixed species flock diversity or potentially flock size improve food finding in a wild songbird community? And we've known for quite a long time that there are serious benefits to being in larger groups related to, anti-predator benefits, lots of other benefits, and also food-related benefits. So there's increased food-related problem solving in house sparrows, in European tits. This is a commonly seen outcome in a wide range of different species. So there should be benefits of being in a larger group. There's also some scattered literature out there, and it's starting to grow more and more, that there's benefits to being in diverse groups. And so Jones in 2004 found that genetically diverse honeybee colonies regulated hive temperature better than homogeneous single-sired honeybee colonies. This has been known in different human groups for decades, that the greater the diversity in human groups, the more effective decision-making those groups can make. This was a study that came out not too long ago in Science, looking at the relationship between diversity in human groups in an experimental setting, and whether groups made better economic decisions. And short story is that yes, they did. And so that raises an interesting question for us in thinking about mixed species groups, because it's not necessarily clear that in a mixed species group you would make the same predictions about those benefits, right? So mixed species groups are ubiquitous in a wide range of systems. They're certainly common in our area: chickadees, titmice, cardinals. Why it's not a given that you'd expect benefits of size or benefits of diversity is that there's often competition among the species in these mixed species groups. There's often clear size-based differences in dominant status within these groups, and so subordinate individuals may be paying a real cost of being in these groups. And so again, it's not necessarily clear that we would necessarily predict the same benefits of being in these mixed species groups, as seen in those earlier studies that I showed. So our mixed species group for this study was Carolina chickadees, which I've been talking about, tufted titmice, and white-breasted nuthatches. In our area, chickadees and titmice are considered nuclear species. These are like the core group members around which other species will associate into relatively stable mixed species flocks. White-breasted nuthatches are one of those kind of outside associating species called a satellite species. Sometimes these guys are present in mixed species flocks of chickadees and titmice, but not necessarily. Chickadees are nuclear species, but they're also by far the most subordinate species in these groups, because they're like half the size of the other group members. So titmice and nuthatches are much more dominant over chickadees. 19 times out of 20, if there's a heterospecific agonistic interaction at a feeder, it's a titmouse supplanting a chickadee, or a nuthatch supplanting a chickadee. So the study I'm going to talk about took place at over 40 different sites in eastern Tennessee. We make sure to keep each site at least 400 meters separated from the next closest site, just for statistical independence. This distance helps ensure that we're measuring different flocks. At 34 of these sites, we had 2 or more species present for this study, and then at 12 of the sites, it was just a conspecific only flock. Only one species was present. And the criterion for inclusion in this particular study was pretty straightforward. We needed any sort of group. We just needed 2 or more individuals. And for brevity today, I'm just going to focus on the chickadee side of the story. So here's the problem that we put them to. So we have these, we call them feeding stations, but that makes them sound more sophisticated than they actually are. It's just a platform feeder, 46 of these spread around different major study locations in eastern Tennessee, steel pole with a wooden board on top. And over the decades, we have used these both for doing studies at feeders where we can present predator stimuli and whatnot, do playbacks, see how birds respond. But we also can set treadle traps on top of these feeders and then catch birds for our aviary studies, or to band them for individual identification in the field. So the birds have gotten used to these platform feeders as a place where, every 2 weeks, there'll be 50 grams of sunflower seed and safflower seed put on them. They don't really predict, they can't predict when we stock the feeders. We're not supplementing them to such an extent that they come to rely on them. We just want the birds to know that these feeders are an unpredictable, but if there's seed on the feeder, a highly, a really important food source for them. So they've gotten used to coming and going from these feeders. For this study, when we saw birds at a feeder that we had just stocked, we would quickly set up the recording equipment, video recorders, audio recorders, and then introduce the problem. And this is a motivational problem rather than a technical problem, because there wasn't really any sort of apparatus that they had to manipulate. What we introduced was a simple hopper feeder attached to the platform feeder that they had been using. Importantly, I think you can see this. You can see this bird seed in this hopper feeder. Birds that are perched on the surrounding cover around the feeder, if they perch on the platform feeder itself, they can easily see the seed that's in this hopper feeder. And the question is, will they overcome their fear of novelty, their neophobia? So it's a motivational problem they have to solve. Will they overcome that neophobia to access the seed? So we had the novel feeder in place for 30 minutes. The data that we collected were super straightforward. So it was the latency for each species to take a first seed, and then seed taking rates for each species. We were able to collect inter-observer reliability at 15 of those 46 sites, and these are really easy data to collect, really high reliability. Chickadees solved the problem at 21 of the 40 flocks where chickadees occurred, right? So roughly half the time, chickadees went and took at least one seed from that novel hopper feeder. And so we collected a series of flock-related measurements to look at both group size and group complexity. So total flock size, this is the total number of these 3 target species. We used a modified Simpson index from ecology to look at what we call the diversity index. So this is the proportion of chickadees, proportion of titmice, proportion of nuthatches. And this ranged from 1 to 3. So the smallest diversity index happened when there's only one species present. The largest diversity index was possible when all three species were present and in equal numbers, equal proportions. And the modal flock size for this was two members of each. We also looked at conspecific group size. So for chickadees, that's simply the number of chickadees in the flock. Heterospecific group size for chickadees, that's the number of titmice and nuthatches. And then the proportion of conspecifics in the flock. And you're probably thinking, boy, these are some seriously correlated measures. So what we did is a principal component analysis to try to reduce the dimensionality of the data set here. And so the first PC score explained about 60% of the variance, and this, we argue, mapped more onto the diversity of the flock. So it's PC1 scores were negatively linked to the proportion of conspecifics, and then positively related to the number of heterospecifics in that diversity index. And then the second principal component explained about a third of the variance. That mapped onto the number of conspecifics and the total flock size. So what did we find? Well, we did a two-step statistical analysis. We used binary logistic regression with those PC scores, looking first at the success or lack of success of the flock at solving that novel feeder task. And then for successful flocks, we did use linear regression with those scores to assess whether there was a relationship between the diversity or the size of the flock, that PC score, and the latency to solve the problem and the relative number of seeds taken. So first, the first step in the procedure. So 19 unsuccessful chickadee flocks, 21 successful chickadee flocks. That first principal component score with flock diversity, what we found is that the chickadee flocks that were successful were significantly more diverse in terms of mixed species flock composition than chickadee flocks that were not successful. And then if you look just at those successful flocks, there was a significant negative relationship between flock diversity and the latency to solve the task. So more diverse flocks were solving the novel feeder task quicker. And then a positive relationship between the diversity of the flock, mixed species flock diversity, and the seed taking rates per chickadee. So this is adjusted for flock size of chickadees. And so the more diverse the flock was, the more each individual chickadee was able to exploit that food resource. What about flock size? Well, there's a tendency for the successful chickadee flocks to be in larger flocks, but it's not statistically significant. And then if we look at just those successful flocks, there's no relationship between that second principal component score linked to flock size and their latency to solve the problem, or the number of seeds taken per bird. So the more diverse the flock was for chickadees, the more likely they were to solve this motivational problem, this novel feeder task. For successful chickadee flocks, furthermore, the more diverse their flocks were in terms of mixed species flock composition, the shorter the latency to solve the problem and the greater their seed taking rates were. So individuals were benefiting from being in more diverse mixed species flocks. And then this asterisk is here because we found this same relationship here for titmice. So the more diverse titmouse flocks were in terms of mixed species flock composition, the more likely a titmouse was, at least one titmouse was, to solve that problem. But for titmice, we didn't find this relationship for those successful flocks. So wrapping up these three studies, what we noticed is really powerful influences of the immediate social group on individual behavior. So just like human social psychology has revealed for our behavior for decades, the immediate social context in lots of animal species plays an important role on individual decision-making and individual behavioral production. And that's certainly the case that we're finding with chickadees and titmice. So what the studies I presented here today showed that group size or maybe group density, variation in that affects the complexity of calls that individuals produce. Taking away variation in group size or density, simple familiarity of group members affects calling in both threatening and movement-related contexts. And then thirdly, diversity in mixed species groups can increase problem solving in novel food finding tasks. So this social psychology consideration extends beyond communicative behavior to at least the behavioral system of ingestive behavior, food finding. So what are some of the next steps? Well, Brittany Coppinger was one of the students I mentioned earlier. She has, for her dissertation research, which she's wrapping up this next academic year, she's done two massive studies, one that's pretty much completed and we're analyzing, writing up the findings, where we manipulated chickadee and titmouse flock composition and density in these aviaries. So these are the 27, I can't remember the exact number, different flocks of titmice and chickadees. Some were only titmouse flocks, some were only chickadee flocks, some were mixed species flocks, a lot of variation in flock size and mixed species flock composition. And we looked at variation in their calling rates and note compositions of calls, both in ambient, just sort of baseline conditions, and also in response to threatening stimuli. We presented them with more problem-solving tasks, both motivational and more technical, where they had to manipulate the feeder, manipulate some apparatus to gain access to a food they really wanted. We have some personality-like measures on the bird, so we're looking at the role that those might play in variation in behavior in these different social contexts. And I'm super excited to say that just a day ago, we got the news that the first big study that came out of Brittany's work has been accepted at Animal Behavior. So we found pretty big heterospecific audience effects on calling behavior in chickadees and to a lesser extent in titmice. So be on the lookout for that. Hopefully that'll come out later this year. And other than that, there's just a lot of folks and a lot of organizations to thank who have contributed to this work over the years. And so those are shown here. And with that, I guess I will wrap this up. Thanks for listening.